everyone should be explaining hearing to their neighbor. Okay, questions? Does it make sense? Could you explain all of these? Let's do an eyes closed exercise. Close your eyes. Everybody close your eyes. Close your eyes. Everybody close your eyes. I'm seeing some eyes open, okay? Okay, eyes closed. Okay, raise your hand if you feel like I could use more explanation of this. Okay, hands down. Some eyes were still open. Okay, you can open your eyes up again. That was 2/3 of the class. Okay, any questions? In the back, yeah.
So I understand the general concept that like specific portions of the cochlea will only have like certain frequencies going through them, but I'm confused on how like the variation in thickness and thinness allows it for only like only on specific sections.
Yeah, so what creates that specificity?
Yeah.
What creates that specificity? Okay, we always unroll the cochlea so that we don't have to deal with that spiral. Okay, so I've got it unrolled up there. So what creates the specificity? What's happening is that imagine that we have a cochlea right here, okay? And we have the two windows. They're the only flexible part. It's encased in bone, so the only flexible part are these two windows. So it's like having a solid structure with rubber sheet here and a rubber sheet here. If we push in here, we create pressure. The only thing that can happen is bulging out of the rubber sheet down below, okay? So we push in on the top one, that bottom one has to bulge out. Okay, remember that a pressure wave is a bunch of molecules that are moving along that are closer together. So it's a bunch of molecules that are closer together and they're moving fast, each new one, so the molecules here are moving fast and hitting the next molecule, okay? So it's as if I came here and I pushed a few of you and you immediately turned around and pushed the next most close people and you immediately turned around, you can join in if you want, and pushed the next. That would be a pressure wave moving across, okay? Or like a wave when people are doing the wave cheer in a stadium. That's a pressure wave. And that pressure wave, when it hits something solid, pushes against it. It pushes in that window. Okay? Frequency is how many of them are coming per second. So if we had 10 per second, okay, that's a low frequency sound. High frequency sound, we hear up to about 20,000. Well, many of you do. I don't. My upper limit is now around 15 or 16,000. Too much loud music when I was younger. Okay? So those pressure waves are hitting, and they're coming at a different pacing. Okay? So as these pressure waves move in, they hit that window, and then they have to cross that intermediate pair of membranes with the hair cells in between, right? They have to push across, so they have to bounce it up and down, right? Each pressure wave has to bounce it down, and then it comes back by elastic recoil. Okay? So it bounces down, and then it has to pass out and push out in that other rubber elastic, not rubber, but elastins membrane. Okay? Okay, so any one, if you only had one pressure wave, you would be thinking about it correctly as it's just going to move that thing. And it'll move it maybe not equally all the way along, but it's just going to move the thing up and down. Okay? The thing about sound is that it's repeated. That's what a frequency is. It's repeated. It's 10 times per second, exactly equally spaced. So a frequency of 10 hertz is 10 per second. Every pressure wave is 100 milliseconds apart. If it's 100 hertz, splitting them up to, what is it, something like 40 milliseconds. Okay? So what you get, instead of just one push, you get these repeated pushes. And the structure of that membrane is such that you form a standing wave. So do you remember those physics high school experiments where you bounced a spring? How many of you did those? Okay. Or when you played with the jump rope ever and bounced it up and down. So there's two ways you can do it. You can do a traveling wave in which the wave goes up and it moves along. Okay? Or you can do a standing wave in which each spot just goes up and down. So the wave looks like it's doing this. Okay, so a traveling wave is moving along. A standing wave has each spot just going up and down. Does that make sense to you? If not, find a video on standing waves on the web. There's tons of them. Okay. Okay, so that standing wave means a particular frequency bounces only one spot up and down. A low frequency has a lot of time between pulses, so the lowest frequencies bounce it up and down here and nowhere else. Okay, so only here. So this might be somewhere at the low end of our range. Right at the end would be around 20 hertz. A really deep sound I can't do. Okay. And here's a really high sound I can't do either. Okay, so 20,000 hertz, that doesn't have time to get all the way down there. It only has time to get here and bounce this up and down. Okay? So the frequency is the location along here, and it's because those frequencies are setting up standing waves. Okay? Okay, another question. Yeah, is that why people with hearing loss can hear? Your comment was lower frequency, and that's common. A lot of times we get damage at the high frequency range, but it doesn't have to be. It can be anywhere. And it's because you have damage to particular areas. So one of the ways you can do that is a really loud sound. We're going to come back to talk about that in a minute, but first I want to take other questions, so hold that thought. Uh-huh.
I get the frequency difference, but amplitude is...
Okay, what about amplitude? So the difference between frequency and amplitude. So frequency, okay. Any more questions about frequency? Uh-huh. Carolina? Right, so part of the reason you get these nice, precise standing waves is because the characteristics are a little bit different in different locations. The physics of an ear are actually really complicated, but that is part of the reason. And you don't need to remember all the details, but I do want you thinking about the fact that, okay, you're creating standing waves, so a particular frequency is only making it bounce up and down in one location. Okay? Okay, so back to Jessica's question about loudness. So when this bounces up and down, we have a membrane on top of these hair cells. So when they go up and down, they go sideways a little bit, right? So every up and down movement bends those hairs, and those hairs have stretch channels between them down near the base. And so bending is going to open stretch channels, sodium stretch channels. So sodium comes in, and you get neurotransmitter released to these neurons I labeled A, B, C, and D. Okay? Okay, so that's how you get the signal for frequency. What about loudness? Well, think about what happens. A quiet sound would be if I pushed you guys at this end just gently, and you reached around and pushed the next person really gently. And it got to the far end of the room, and it was just a gentle push against the wall, which is going to give you only a small up and down movement here. You're only going to open a few a few sodium stretch channels, not a lot. And with only a few, you only release a little bit of neurotransmitter, which brings only a little bit of sodium into cell A, and very few action potentials per second. Okay. So if it's a quiet sound, you get few action potentials per second. What about a loud sound? Well, loud sound is me pushing more violently. So I push you guys really hard, you turn around and you slam into the next person who slams into the next person. And at the far end of the room, we're ramming into the wall. Okay? A very violent push here. So what happens to this membrane? Do we still have a very small movement? No, we've rammed it down. Okay? So now we have violent movement. A loud sound gives you violent movement. What do you suppose happens to those stretch channels? They stay open longer. So you get more neurotransmitter release, and you get more action potentials per second. That's a loud sound. Okay? Okay, another piece I want you thinking about, you have an eardrum here. So here's the tympanum. And you have amplifiers, a series of bones, which I'm not even going to try to draw. 3 bones or ossicles, 3 middle ear bones. And they're a lever system that amplifies the movement. So a relatively small movement of the tympanum winds up being a bigger movement, bigger deflection back and forth over here. It amplifies the sound. That helps you hear much quieter sounds. But haven't we created a problem? What if you hear a loud sound? Don't you risk damaging, tearing that membrane? Or moving this membrane, the tectorial membrane, up and down so rapidly that you start damaging those cells? And that, it turns out, is a very serious problem. So you gain amplification, but you risk greater damage to your ears from a loud sound. Really violent movement of this up and down, because you're pushing so hard on these hairs, you can tear the cells and kill them. So loud sounds risk tearing those. That's the reason I don't hear in the high frequencies anymore. Okay. So how do your ears deal with that? You have a muscle . Has a name, but I'm just going to call it a dampener, a sound dampener, sound quieter. You have a muscle, and there's a neural circuit that if there is a really violent movement of the tympanum, I'm running out of colors. Let's see. Let's go with green. So if there's a very violent movement of the tympanum, a really loud sound, that activates, so stretch channels in here, activates this sensory neuron, which goes to a motor neuron, which contracts this muscle. That contraction minimizes the force that's transmitted to your cochlea. So that dampens the sound, reduces the violence of the movement. That turns out to be an extremely important muscle. It's the reason why you can hear a thunderclap, a really loud sound, or an explosion nearby, and most of the time you have no ear damage if it's not too close, not too violent. It's a very fast reflex, and so very quickly that muscle contracts, dampens the sound, and you protect your hair cells from damage. Okay, make sense? Okay, so, yeah, go ahead, Christian. So ringing sounds in your ears can be from, and I don't know that it's known exactly what's causing it, but you're getting some, potentially some damage, potentially it's neurotransmitter depletion, I'm not quite sure. But the ringing sound indicates you were either close to damage or you caused damage. Ringing sounds after loud noise are not a good thing. Okay? They're a hint. Okay, so one last point on this, then I'll take another question. This muscle and this reflex you have no conscious awareness of. You never know what it's doing. Completely unconscious reflex. This muscle can fatigue. Fatigue meaning it runs out of ATP. It can't replenish its ATP fast enough, so it fatigues and is no longer able to contract. So what's going to happen if you're in a loud sound environment for a long time? Tell your neighbor. You're in a loud sound environment for a long time. So what happens? You're there for the first 20 minutes, things are okay. Maybe the next 30, 40 minutes, things are okay. Somewhere around in that time, this muscle starts to fatigue. And now that pounding sound, I'm remembering some specific concerts when I was a kid, that pounding sound is now not dampened. That muscle is limp. You pound here, and you're tearing now those cells, and you don't have any conscious awareness that you're doing it. Same thing happens, earbuds that are loud enough, same thing happens. You are not aware that that muscle is fatiguing, and so you are not aware that you're causing damage. Okay? So tell your friends, keep their sound down. By the way, if you walk by somebody and you can hear their earbuds from that far away, that is loud enough that they are quite likely doing unpleasant things to their ears.
It depends on the earbud quality, though. Like, some of them are, like, shaped weird.
So it probably does depend on the earbud quality, but I couldn't tell you how. It depends. But I can tell you, if it's that loud, it is not a good thing for your ear. High quality sound that is too loud is still too loud. Okay. Uh-huh.
Why would the weakening of the muscle not cause you to hear a difference in the sound?
So why would the weakening of the muscle not cause you to hear a difference in the sound? So this has to do with something we're not covering in this class. I do talk about it in physiology, adaptation to sound. What happens is there are many neurons adapt to the frequency, to the amount of stimulus they're getting. And so initially, you get, when a loud sound starts, you get an increase in action potentials. But if it stays, those neurons adapt and they drop back down to a lower level of action potentials per second. So for hearing, you don't actually hear the actual sound. You hear the relative sound. How has it been changing? So your ears adapt, and you're no longer aware that it's a really loud sound, even though you're damaging your ears. And that's the problem. When it comes on suddenly, you'll hear it loud and painful. If you go into a really loud environment, check this for yourselves. You can go to a really loud place, and initially it's hurting your ears. And then you may notice 5 or 10 minutes later, maybe even faster, it's not doing that anymore. That's partly because of adaptation. The sound probably is too loud. If it was hurting your ears initially, it's probably too loud. Okay.
Most really loud sounds are, like, lower pitched. So what could cause, like, it seems like it's more common to have damage.
Seems like most loud sounds are lower pitched, so what would cause damage to higher frequencies? I can't answer that question completely, but I can tell you that there actually are some sounds that are really high-pitched and really loud. Think of a high-pitched scream. And ultrasonics, so things at the high end of our range, like steam whistling out of an opening or something like that, there are some really high-pitched sounds that are quite loud. For reasons that I couldn't explain, we tend to be more sensitive at the high end. Okay, let me move on to the next thing. And I want you all to remember that when I say, do you have any questions? If you have a question, there's a good chance the other 2/3 of the class also has a question. Okay? There really were 2/3 of the hands up, and none of you asked the question before I told you that. Which I understand, but I want you to believe it's worth asking those questions. Okay, I gave you a really tough question about rhodopsin and how those different opsins could potentially detect different wavelengths of light, different photons. And for that one, what I hoped you got out of it, I don't know if it actually worked, what I hoped you got out of it was that if that first of all, a regulatory mutation isn't going to do you any good, because it's exactly the same protein, there's just more of it or less of it. So that shouldn't respond differently to different wavelengths of light. So that means you need a coding region mutation, and I also wanted you to think about where would that coding region mutation be. Well, a coding region mutation, for example, near the G protein isn't going to help you much, because that's down with the area where it's affecting the G protein. It's not responsible for detecting wavelength. So let's think about structure of a protein, and this is partly about rhodopsin. It's partly about you thinking more deeply about how the structure of a protein is related to function. Okay, so I'm just going to draw some squiggles here. And I'm leaving a space in the middle. I'm leaving a space in the middle that is going to be the binding pocket. So that's where we're going to put retinal. Okay? Now remember, this is a sequence of amino acids. I've made the binding pocket bigger than it really is. Retinal's a little smaller than this, quite a bit smaller, but we wouldn't be able to see it very well. If I tried to draw it with the binding pocket the right size. Okay, so here's rhodopsin with its G protein. And if you wanted to change the interaction with the G protein, so mutations that change an amino acid here they'll alter G protein activation. Make sense? If you want to affect something having to do with absorbing photons, you have to do something that's up in the area of the retinal. So the mutations likely mutation sites to alter photon absorbance. They're going to be up there around the binding pocket. So what are the things you might be able to do? Well, I can think of two possibilities. There might be more. One is that maybe the angle or tension of retinal affects the likelihood of a particular photon being absorbed. Photons have different wavelengths. We know that the iodopsins have different sensitivities, so that tells you it must be possible to have some wavelengths be absorbed and others not. So maybe the way the retinal is held, the angle, affects the photons that are being absorbed. Could you potentially have a mutation that changes an amino acid so that instead of being oriented this way, remember retinal is a ring with a tail and a bend. So instead of being oriented this way, it's oriented this way. Could a mutation change the orientation? Okay, that's one thing you could do and propose, okay, maybe the angle at which the rhodopsin is held affects it. Maybe the binding pocket puts a little bit of tension on that 11 cis bend. And maybe the tension on that 11 cis bend affects how easy it is to absorb a photon of a particular wavelength. Again, a mutation in the binding pocket. Another possibility is that part of the protein absorbs some photons and lets others pass. If that's true, turns out it isn't, but it's a perfectly reasonable hypothesis. If part of the protein absorbs a photon of particular wavelengths, maybe a mutation between where the photon is coming into the protein and when it reaches the retinal, maybe a mutation there would actually change which photons can pass through. And that would be a very reasonable answer. Okay, back.
What's the difference between a metabotropic receptor and a G protein coupled receptor?
Okay, so what's the difference between a metabotropic and a G protein coupled receptor? An important question. A G protein coupled receptor is one category of metabotropic receptors, and it's the one we're using over and over. Another major category are tyrosine kinase receptors. Those are also metabotropic receptors, but we don't talk about them because we don't need them conceptually. Once you understand how G-protein coupled receptors work, you understand in principle how these others do. And there's more types of metabotropic receptors. So G-protein coupled receptors are one type. The reason I chose those is that for every system we use, many of the metabotropic receptors are G-protein coupled receptors. If you have reason to think something is a metabotropic receptor, your first guess ought to be G-protein coupled receptor. And then some of those other categories, okay? Okay, so mutations in those areas then somehow, and I don't know how, I don't actually know the answer to this question, are changing the likelihood that a retinal will bind a photon of a particular wavelength. And that's color. That's how we get our sensation of color. So the cycle is, so the question is, can I just go over again the mechanism really quickly? Let's have a photon coming in. So here's a photon. Why am I drawing this wiggly line? These photons have a wavelength, right? Are they particles or waves? I don't know. That's one of those physicists' metaphysical questions. But okay, so it starts off as, I think I only drew 5, but it should have 6. So here's our 11 cis. Our 11 cis bend, okay? And when the photon hits, it straightens out. Now it doesn't fit in its binding pocket anymore. And we have all trans. Trans meaning straight. Cis is a bend. Okay. Okay, so all trans retinal. That activates the G protein, but it activates the G protein briefly, but this receptor is now out of commission, does nothing until it gets an 11 cis retinal, a bent one, back into its binding pocket. That is part of how you adapt to changing light levels. So go from here, which is comparatively dim, out into bright sunlight, and within seconds, you have moved retinal from being 11 cis retinal. In fact, when you go out after this class, I want you imagining this. 11 cis retinal, maybe 90% or more of your opsin molecules have had the retinal pop free. And it's going to be 20 minutes on average, probably a little less than that. But anyway, 20 minutes or so before retinals get the bend and pop back in. Okay. So once they come out, the retinals can lose the energy of the photon, convert back to 11 cis, and then they can go back into a retinal. Back into a rhodopsin. Okay? Does that make sense? Okay, so that's the cycle. Now I want us thinking about eyes, mammalian, bird, reptilian, so we're talking about chordate eyes. Similar principles would apply in others, but we know chordates much better. And remember again, difference between an eye spot, some cells that have these sensory receptor proteins, right, that have rhodopsins in them, versus an eye cup, which gives you direction, versus a vesicular eye, which gives you a true image. So higher chordates have vesicular eyes, so do mollusks, so do some jellyfish, independently evolved. And that gives you the capacity to process that image to detect shapes and patterns and colors. So there's ways to adjust light intensity that include the size, the size of the retina, how big your pupil is, not the retina, how big your pupil is. So are you closing something to cut off a lot of the light, or are you opening it back out? Okay, so that affects how much light you get. That's part of how you adjust to bright light. In bright light, you decrease the diameter of your pupil to reduce the light coming in. A much bigger part is what I just talked about, the retinal being dissociated from rhodopsin, and then there's another molecular mechanism that also helps you adapt to bright light. The light comes in, it hits those photoreceptor cells in the back of the retina, and we detect not just patterns, but also colors. In bright light like this, you're using mainly your cones, red, green, blue cones. In dim light, you're using your rods, which have a broader sensitivity, most sensitive in the green, but broader sensitivity, and they are more sensitive to dim light, but they don't give you any color. Okay, just a single broad presence of light or not. Your iodopsins detect light, but many vertebrates, in fact, most vertebrates, have iodopsins you don't have. So the iodopsins are the ones in your cones. You have 3 cones. Each cone expresses one of the 3 iodopsins, red, green, or blue. And you detect color by the combination of how much a little spot of a red, green, and blue are stimulated and how brightly the red is compared to the green compared to the blue. If only the blue is stimulated, there are no red or green photons, you detect it as blue. If red and green are stimulated equally, you detect it as something in the middle, purple. Okay, make sense? And you could do neural circuits, right? For purple versus red versus green. Do you agree? Okay, but our system is not that good. Birds detect in the ultraviolet. We do not. So if you use something to assess the ultraviolet absorbance of these flowers here, what we see is this yellow flower with a black center. What a bird sees is additional color. This is only shown in grays, but they're actually seeing these as additional colors that you cannot see. Here are the mammalian visual pigments, and these are the iodopsins. So rhodopsin is your one for night and dim light vision. The iodopsins, notice where these peaks are. If you were trying to get maximum sensitivity to color, wouldn't you rather have a peak here? Doesn't it seem like the green and the red are awfully similar and overlap an awful lot, so that a red will actually stimulate, most red photons could stimulate both, and most greens could stimulate both. Might it be nicer to have the curve look more like that? And the answer is yes, it would. That would be better. So why do we have this crappy green, which is sadly the truth? You have an inferior green iodopsin. I'll let that soak in for a minute. Deeply inferior. Let's look at what birds have. Here's what birds have. So before we were looking at, the figure only went down to 400. Okay, so before the line was here, that was our axis, this black line. So well below where we detect anything, birds have this ultraviolet. That's what I just showed you a moment ago. But look at where their green is. So your green your peak is here. But for birds, it's evenly spaced between the blue and the red. They have a nice green. They have a good green. We have a crappy one. Why? Well, the problem is we lost 2. If you look back at ancestral animals, the phylogenies indicate very clearly when you look at outgroups. The phylogenies indicate really clearly that the ancestor of birds, early birds, and modern birds all had an ultraviolet, a blue, a green, and a red, all evenly spaced, just as you would hope they would be to work well. And that's true, in fact, even if you look for ancestors of mammals and birds. The common vertebrate ancestor here had all 4. But what happened in early mammals is 2 were lost. Why? Why lose 2 perfectly good cones? So we lost the ultraviolet and we lost the green. We kept the blue and the red. Why? Well, early mammals were apparently under strong selection to be nocturnal. Color vision isn't that useful at night. The problem with color vision is it takes at least 3 cones to get the same amount of information you can get from one rod. It takes more space to get the same information. And if you're under strong pressure to be able to detect that nocturnal predator, you want the brightest detection you can get. So selection strongly favored more rods, not just for space in the retina, but also for processing in your brain. And as that selection put pressure on favoring night vision, at some point, your color vision is actually going to be better by losing a cone and having just 3 and eventually 2 remaining, because you don't have enough processing power to process all those different colors. So selection favored night vision and favored losing these 2 cones. Most mammals still only have those 2. Primates, very diurnal, active during the day, primates regained one of them. An early Old World primate had a gene duplication of a red. And then mutations. Where in the protein would those mutations have been? Down near where it interacts with the G protein? Nope. Okay. Up in that region that's holding the retinal, whatever it is that's making it sensitive to different wavelengths. But there hasn't been time for that much evolution. So selection has been moving it, but it's not moved very far yet. So selection favors moving that green more toward the center, but we're not there. So we inherited a nice blue, a nice red, and a crappy green. In principle, we could get them back. People have taken the green cone, the gene for the green cone, and inserted it into mice. And if you do that, what they do is instead of producing 2 cones, just a red and a blue, scattered all over their retina, they produce all 3. And each one properly only expresses 1 iodopsin. And now you have created a mouse that has better color vision. We, in principle, could take a bird green cone, destroy our crappy green, put it into an embryo, and have that embryo now have a good green and have better color vision than you do. And in fact, in principle, we could take the ultraviolet cone and give it to humans as well and see far more colors than you can see. Look around you. Every green you see would probably look really different. Every yellow, every purple would probably look really different because it probably mixes wavelengths in different ways and may absorb or reflect ultraviolet, in which case you would see something different. We could give ourselves, or our children anyway, you can't do it, but we can give our children much better color vision, who then will grow up and say, Mom and Dad, I do not understand your color sense. What made you think these 2 colors match? And you're looking at them saying, they're both green. What do you mean? They're a perfect match. And the kid is saying, they're not even close. Okay, so here's an ethical dilemma for you. If you had the chance, eyes closed exercise. If you had the chance, would you give your kids a better green? Okay, close your eyes. Close your eyes. Everybody close your eyes. Close your eyes. Close your eyes. Some people's eyes are open. Close your eyes. Eyes closed. Okay, raise your hand if you would give your kid a better green. Okay, keep your eyes closed, hands down. Raise your hands if you feel like this would be a bad idea. Color menace. Okay, hands down. You are not quite 50-50 divided. About 40% of you would give your child the better green, at least today, hypothetically. Okay, a couple last things before we move on to the endocrine system. I should have known I wasn't going to get very far into endocrine system today. You may have noticed these dotted lines versus the solid greens. Another thing we have lost is the ability to narrow those wavelengths. So the dotted lines are the sensitivity to wavelengths of just the isolated protein, the iodopsin. The solid lines are what a bird is actually detecting. And that's because above each cone in a bird eye is a little oil droplet. And those oil droplets are colored. They absorb some photons. And different iodopsins have different cells, they have different oil droplets. And so it filters out some of the lights, and so that makes these even better. Notice the blue and the green overlap only a tiny amount in the solid line, whereas they overlap much more with the dashed line. And the red and the green overlap much less with the solid line. So not only do they have better iodopsins than you do, apparently mammals do have some of these oil droplets, but they don't do the color filtration. So we don't have that either. Not only do you have a crappy green, you don't have the oil droplet filter above it. I'm going to move on, so catch me with that question after class. Okay, so birds, much better off. Lizards are better off than you are. Next time you look at a lizard, I want you to think about its eyes and ponder your own inferiority. I want to make another distinction, nociception versus pain. So this is a map showing some of the pain processing pathways in which we start with damage from a pain source. Stab yourself with a pin, don't do it. Okay, and here's the sensory neuron. There's the connection in the middle here to an inner neuron that's going back up to the brain. So sensory neuron back to the spinal cord. Notice it says dorsal root ganglion up at the top left. Ganglion's a cluster of cell bodies outside the central nervous system. So that runs up to the brain, goes through the medulla, periaqueductal gray, who comes up with these names? Runs up into the thalamus. Remember I talked about the thalamus as an important relay system. Thalamus, and then out to the cortex. Notice all the places it's going. So we're getting these pain pathways going to multiple locations. And they're doing different things. They're potentially causing different kinds of pain. Some of these are circuits that you're not consciously aware of. So some of these are causing events that are potentially causing changes in your brain that you don't have conscious access to. It's perfectly possible. In fact, in animals with no higher consciousness, so if you think about a nematode with its 256 or so neurons, we know what almost every single one does. About a quarter of them are sensory, a quarter of them are motor, something like a half are interneurons. There's nothing left over for consciousness. They're just a bundle of reflexes. So they don't have any reflex, they don't have anything except reflexes, but they still respond to damaging stimuli. How do they do it? Well, it's just a reflex circuit, right? You cause the damage, and that triggers muscle movement to get away from the damage, from whatever's causing the damage. And so they'll move away from acid, they'll move away from heat, they'll move away from a stab wound, okay, through these reflexes. Pain becomes useful as animals get to the point where they can learn. Pain is a teaching tool. That's one piece. It's a teaching tool that says, don't do that again. Okay? That's how I learned not to put my fingers in the electrical socket a second time. So pain is a teaching tool. Second is, it's an indicator of damage. Don't do things that make this damaged foot be damaged any further. Give it a chance to heal. Okay? So pain does those two things. Helpful for an animal that is conscious because it can affect decisions, and that animal can also make decisions like, well, yeah, I know it's going to cause more damage, but that's a tiger. I'm running anyway. Okay? Okay, I want to spend the last 5 minutes just getting started on the endocrine system. This is what we're doing for homework last night. And we've been talking about neurons and sensory systems. A problem with those is that anything you want to do with a neuron, information comes in through sensory systems, goes to a neuron, you have to send that information to every individual cell with a physical structure, an axon. So any communication you want to do requires a lot of resources. You have to put resources into that branch of an axon. But what if nearly every cell in the animal has to change, going from a tadpole to a froglet to a frog? You can't send an axon to every single cell in the skin and to every single part of the skeletal system. You can't do that. It's way too many resources. So hormones are a way to reach many, many cells at a little bit slower pace. You've got to wait for the chemical signal to be circulated through the body. As you might expect, animals with circulatory systems tend to use a lot more hormones because they can move them around through the whole body. So hormones are a cheap but efficient way to get messages all over the place. They use the same kinds of receptors, metabotropic receptors and ionotropic receptors. They add a category that we've talked about in only one way. We've talked about transcription factors that can bind a second messenger, move into the nucleus, and change transcription. We talked about it for development. One category of hormone receptors is transcription factors that bind to a hormone that actually gets into the cell, so hormones that can cross the membrane, usually cholesterol-derived. Cholesterol is a membrane component, bind to the transcription factor, and then the transcription factor does something. So we're going to talk about water-soluble hormones that are binding to surface receptors. Same ones. Some neurotransmitter receptors are also hormone receptors. Okay, some neurotransmitters are also released into the blood as hormones. And then we're going to add transcription factors as receptors. The one that was in your homework and that we're going to talk about much more on Monday, the thyroid hormones. And you don't have to memorize any of these hormone structures, but I did want you to just see them. And here's our iodines. And if we had an iodine here, then it would be inactive T4. But with just these three in these positions, this is T3, or thyroxine. Okay? Many hormones are amino acid derivatives. It's useful often. You're going to keep your hormones cheapest if they're small molecules. And so take an amino acid, change its structure a little bit so it'll bind to a receptor, and you've got a hormone. So here's the amino acid, tyrosine, and change it a little bit, just out here and here. Oh, we've also taken off the amino group. Actually, we've just shortened it. Okay, change it a little bit, and we have the hormone epinephrine. You don't need to remember the structure, but I want you realizing, oh, that makes sense. Change an amino acid a little bit, and when it binds to a receptor, it can be a signal. Here's one of my favorites, tryptophan. It's in your book. I'm glad they put it there. Tryptophan, another amino acid. And change it a little bit. We've added this, and we've changed it a little bit on this end, and we get the hormone melatonin. Melatonin is what you're secreting at night, in the dark, and it's telling your body, it's telling cells, it's night. Do night-related things. And during the day, right now, you've suppressed it, and it's telling your body it's day. Some of you are falling asleep despite this exciting lecture. The melatonin is not doing its job properly. Okay, we'll come back next time and talk more about thyroid hormones. And we have an exam coming up in about a week and a half. And it's going to cover through endocrine systems and reproduction. Exams are down front if you didn't pick it up.